Hi friend, welcome to my YouTube channel. In this video, I'm going to discuss on Mitosporic fungi, the status of asexual fungi and their teleomorphic relationship. Mitosporic fungi are also known as anamorphic fungi or residual fungi, or earlier it was known as Deuteromycetes or the Deuteromycotina or the fungi imperfecti. They have some special features like they are septate, they are mycelial fungi, they reproduce exclusively by conidia or mitospores. But there is no record of asexual stage of any Ascomycete or the Basidiomycota. These are also known as unconnected residual fungi, either lost their sexuality or adopted alternative to sex like parasexuality. They are not a monophyletic fungi. The DNA sequencing of these mitosporic fungi makes the residual organism within group of teleomorphic fungi. And the comparative analysis of DNA sequences placed the asexual fungi in the existing classes. The work of Pier Andrea Saccardo, who was an Italian botanist and mycologist who did lot of work during 1880 to 1886, and he grouped these fungi imperfecti into 4 orders: monilials, melanchonials, spiropsidals and mycelia sterila. The organisms that grouped under monilials were of the characteristic feature like conidiophores or conidia. They are directly borne on hyphae. These were further grouped into 4 different type of family: moniliaceae, dematiaceae, stilbelliaceae and tuberculariaceae, followed by melanchonials. And the organism that grouped under melanchonials, here the conidia form in saucer or the disc-shaped structure called acervuli, and singular that is acervulus. And it has one family, that is melanchoniaceae, followed by spiropsidals. Here the organisms produces conidia in flux-shaped receptacles. Those are called pycnidia, and in singular form pycnidium. And it was further grouped into 4 family: Spiropsidiaceae, Zithiaceae, Leptostomataceae and Exzipuliaceae. And last order, that is mycelia sterila. Here the organism's mycelia was sterile or they lacked conidia. And Xylus fungorum was the first system of classifying hypomycetes, where fungus were grouped based on the morphology of the spores. And the fungus were initially grouped into 7 sections based on septation as well as shape of mature conidia. Number 1 is amidospores. Here spores have one celled, non-septate, spherical, ovoid to elongated, and followed by didymospores. Here spores are 2-celled, but they were ovoid to oblong, followed by phragmospore. Here spores have 2 or many celled with septa by transverse septa, followed by dictyospore. Here spores are muriform, that is transverse as well as longitudinal septa are present, followed by scolacospore. Spores are slender, filiform, then several celled, hyaline or colored, followed by helicospore. Here spores are helical shaped, curved allantoid, hyaline or colored, followed by Mitosporic. From the name itself we can say the conidia are star-shaped, striated, radiate or trifarate, hyaline or colored. And these groups were made based on the septation as well as shape of matured conidia, and these are also known as sacchardos spore type. During 1973, Geoffrey Claude Ainsworth, popularly known as G.C. Ainsworth, he grouped the fungi imperfecti into three classes. These are Coelomycetes, Hyphomycetes and Blastomycetes. In case of Coelomycetes, here the fungi grouped under Coelomycetes, their spores formed in Pycnidia or Acervuli. On the other hand, the fungi that grouped under the class Hyphomycetes were further divided into two groups. Sterile hyphae, that means here the fungi mycelia is sterile in nature, they never formed any conidia. And second one is genera bearing conidia on hyphae, where the mycelia will bear conidia. And third class is Blastomyces. All imperfect yeast are grouped under Blastomycetes. During 1910 and 1911, Jean-Paul Vuillemin, who was a French mycologist, he studied various mode of origin of the conidia, and he was the scientist who put stressed that conidia be distinguished by their origin, and the conidia always born on conidiomata. That means these are specialized conidia bearing structure and includes Acervulus, Pycnidium, Sporodocium as well as Cinnamata. During 1953, as a Hughes, he has given a classic classification which is known as Huggesian classification. He put forwarded the scheme of classifying the Hyphomycetes based on conidial ontogeny, that is origin of the conidia, and he divided into four sections representing particular mode of conidial development. And as I said, the system of classification based on conidial origin is also known as Huggesian classification. Now let us see what is that classification based on origin of spore type, which are grouped into blastospores, gangliospores, porospores, phyllospores and arthrospores. In case of blastospores, there is the first group, conidia always born as buds or blown out tip. The common example is Torula and Cladosporium. Followed by gangliospores. Here conidia develop from the swollen tip of the conidiophore, and common example is Scopoliopsis and Amblyiosporium. Followed by porospore. Here the organism produces the conidia that is either in single or whole of thick walled, and these are formed through one or more small pore in sporophore walls. Common example of porospore producing organisms are Alginaria, Carvularia, Mitosporium and etc. Followed by phyllospores. Here conidia born on short unicellular oval or subcylindrical or flux-shaped phyllid with distinct basal swelling and narrow dista. The common example like Aspergillus, Penicillium, they produces phyllospores. And another group, that is arthrospores. Here conidia always formed by breaking of the hyphae into cells. The most common example of the organism that produces arthrospore is Geotrichum. There are a lot of development from 1973, that is Kendrick and Carmichael, who studied a lot and given the classification of the fungi, more particularly Fungi imperfecti, followed by Hawksworth classification proposed by Hawksworth and co-worker 1995, and presently we are following the classification of Kirk and co-worker proposed during 2008. Presently Fungi imperfecti is grouped under 4 different groups. First one is Fungi imperfecti, formerly known as Blastomycetes, followed by Hyphomycetes. Anamorphic fungi, formerly known as Hyphomycetes, followed by acerbular imperfect fungi, formerly known as Melanchonials, and then last group is pycnidial imperfect fungi, formerly known as Sphaeropsidales. There are different conidial shape and septation. To classify the conidia according to the Saccardus classification, and Saccardo made different group, and the groups were named as A, B, C, D, E, F, G. If you see this table, the organisms which are one-celled, these are known as Mitosporic. Already I have discussed in my earlier slide, but they were further grouped into 2 based on whether the conidia are hyaline or bright colored, or conidia were pigmented or dark colored. So suppose group A, one-celled, these are known as Mitosporic. If they are hyaline or bright, these are known as Hyalosporic. And if they are pigmented or the dark, these are known as Phaeosporic. Followed by 2-celled, that is Didymosporic. So similar to group 1, if they are hyaline or bright, these are known as Hyalodidymi. And if they are pigmented or the dark, these are known as Phaeodidymi. Followed by too many cells, that is multiseptate, these are known as Phragmosporic. If they are hyaline or the bright, then these called as Hyalophragmi. And if they are pigmented or dark, then this type of conidia are known as Phaephragmi. Group D, that is Muriform. Here these are known as Dictyosporic. If they are hyaline or the bright, these are known as Hyalodictyi. And if the conidia are pigmented or the dark, these are grouped as or known as Phaeodictyi. Followed by group E, that is Filiform, Scoloascospori. Followed by group F, these are helical, and these are commonly known as Helicospori. And group Z, these are branched. If they are branched, then this has been termed as type Sterospori. There are different type of conidiomartal type as well as conidial spore group. Commonly there are 9 different conidiomartal type as well as conidial spore group. The group has been named from 1 to 9, and these are hyphal, followed by cinematal, then third group is sporodochial, followed by pycnidial, followed by pycnothyreal, followed by cervular, then cupulate, stomatic, and sclerotia. If you see this flowchart, then it will be clear about the classification of the fungi imperfecti up to general level. As I said in my earlier slide, fungi imperfecti has been grouped into 4. First group is imperfect yeast. Here the genus Candida, Cryptococcus, and Sporobolomyces are included. The second group, that is Hyphomycetes anamorphic fungi, here large number of genus are generally included, starting from Alternaria, Botrytis, Sarcospora, Carvularia, Fusarium, then Helminthosporium, Lemonaria, Monelia, Nigrospora, Pyricularia, Rhizoctonia, Trichothecium, Verticillium. Followed by third group, that is a cervular imperfect fungi. Here 3 genus are generally included. These are Callitrotrichum, Pestaliopsis, then Sphalocema. Followed by pycnidial imperfect fungi. And the genus included under pycnidial imperfect fungi are Ascochaeta, Lesidioplodia, Diplodia, Phylosticta, then Phoma, Phomopsis, as well as Septoria. Let us see some of the example of anamorphic genera, that is imperfect stage, and their teleomorphic or the perfect stage. So let us start with the genera East. So here I have cited 3 examples. Number 1 is Candida albicans. Its perfect stage is Saccharomycetals, followed by Cryptococcus neoformans. Its perfect stage is Phylobasidiella neoformans. And the third example is Sporobolomyces salmonicolor. Its perfect stage is Azospora salmonicolor. Then let us move to next group, that is Hypomycetes genera. I have cited here number of examples. Number 1 is Alternaria. Its perfect stage is Livia or Comoclathris or Cocciolobulus, followed by Bipolaris Sorokinia. Its perfect stage is Cocciolobulus sativus. Then Botrytis, its perfect stage is Botrytinia. Then Sarcospora, its perfect stage is Mycospirilla and Spirulina. Carvularia, its perfect stage is Cocciolobulus. Dressera, its perfect stage is Pyrenophora. Fusarium, its perfect stage is Gibberella or Nectaria or Calonectaria, followed by Monilia. Its perfect stage is Monilinia. Then Nigrospora, its perfect stage is Cuscia. Pyracularia, it is the common example. Its perfect stage is Magna Borde. And last example, that is Rhizoctonia. Its perfect stage is Thanatophorus. Then the group C, that is a Servular genera. Here I have cited 3 examples. Number 1 is Coletotrichum. Its perfect stage is Glomorella. And then Pestaliopsis. Its perfect stage is Pestaliosferia. Then third example is Specialoma. Its perfect stage is Alcyone. Then last group, that is Pycidial genera. Here the first example is Ascochyta. Its perfect stage is Didymela. Then Diplodia, its perfect stage is Botrysperia. Phoma, its perfect stage is Didymela. Phomopsis, its perfect stage is Diporde. Then Phylosticta, its perfect stage is Guigneridia. And last example, that is Septoria, its perfect stage is Mycospirilla. And the genera as well as the species of the fungi imperfecti, these are connected to any Ascomycetes or the Basidiomycetes fungi, are shifted to Asco as well as Basidiomycota. So with this, I end up the information related to Mitosporic fungi. Thank you for watching this video.